Good afternoon, student. Today we are going to start new topic, that is crop improvement. In that we are going to see how the quality of the crop or agricultural product is improved. So there are several techniques to improve the crop quality. These are conventional, and some of these are newly, like transgenic plant production. So in your syllabus there are two crops are given: FlavrSavr Tomato and golden rice. In today's lecture we are going to see how the FlavrSavr Tomato, which is transgenic plant, which is developed through genetic engineering, or which is the genetically modified plant, and this is the first commercial product which is accepted, or which came into the market, which is agricultural product. So first we will see the background. So major problem of agriculture is spoilage of the vegetables. So spoilage of the vegetables means they have left shelf life, they spoil early in 1 to 2 weeks, right? So that is the one problem, that is the major problem in agricultural, right? It's to perish the ability of its product. As I told you, there are many methods those are used to increase the shelf life of the agricultural products, and that is mainly for the fruits and vegetables. For grains there is no requirement as like fruits and vegetables. So the techniques are used to delay the ripening process of the fruits and vegetables through the genetic engineering. So how the fruit ripening takes place. So first we will see how in the fruits, how the fruit ripening takes place. So you know some background, like there is ethylene, which is which one is plant hormone that helps or that triggers the fruit ripening, right? So what happens other than that? So the chlorophyll start degrade, right? So what happens due to degradation of chlorophyll, the green color will disappear, and you will observe the different color, right? In case of mango you will find yellow color, in case of tomato that will be red color, in different fruit you will observe different color. So in case of tomato, the red pigment you will find, and that is due to the lycopene. So lycopene will give the red color. And what happens, the fruit gets soft, or you will find the fruits gets weak. So that is due to the degradation of cell wall. So how this degradation of cell wall takes place? So there is one enzyme, we are talking about the tomato, keep in mind. So there is one enzyme, namely polygalactourinates. So this is one enzyme, so that degrades the pectin, which is present in the cell wall of the tomato, right? So what happens, you know the in case of plant cell, there is cell wall is present, cell wall contains the pectin and cellulose, right? So that maintain the rigidity of the cell. So due to this enzyme, what happens, the bond between the pectin that get degrades, and it becomes weak. And you know what is the role of ethylene. And then breakdown of starch to sugar, right? Starch which is polymeric carbohydrate that get degraded into the sugar, and that will improve the flavor, taste and smell of the fruit. So this is the ripening process of the fruit. Now we are going to see how this FlavrSavr Tomato is developed. So we will see the genes which are involved in this. The first is pTOM5 gene, which encodes phytosin synthase, right? That promote the lycopene synthesis, that impart the red coloration. Keep in mind, phytosin synthase, and this is one enzyme which promote the lycopene synthesis. Then pTOM6, which encodes polygalactourinates, that is abbreviated as PG. Then these enzymes degrade the cell wall, mainly the pectin, which is present in the cell wall of the tomato cells, and that will result in the fruit softening. Then last gene is PTOM, which encodes ACC oxidase that will trigger the synthesis of ethylene, which is plant hormone, and that will stimulate the fruit ripening. Keep in mind, ACC oxidase that catalyze the ethylene formation, and ethylene will trigger the fruit ripening. So these are the enzymes. Then now we will see how the FlavrSavr crop, which is genetically modified plant, is developed. So what is the characteristics of this FlavrSavr tomato? You know, in normal tomato the shelf life is up to the 1 week. After 1 week you will find that will get soft or it may spoil. So this genetically modified tomato, FlavrSavr tomato, this plant species has longer shelf life compared to the normal tomato, and keep in mind, this is the first commercially available genetically modified food. Then so this species or this crop is produced by one company, namely as Carlgin, in 1992. So what is the technique they have used? They have used antisense RNA technology. In last year you may have studied what is miRNA and what is siRNA, what is their role, right? So to understand this technique you should read what is miRNA and what is siRNA. So the technique which is used by this company is antisense RNA technology. So what happens in that, the antisense RNA, or you can say the mRNA, which is complementary mRNA to the PG gene, that will inhibit the synthesis of PG enzymes. You know the PG genes that will encodes for PG enzyme, polygalactourenates, and it is developed in 1994. So now we will see in detail. So in short I will explain you so how this technique is developed. So you can see in normal tomato which contains mRNA of PG, which is called as sense RNA, then it will be translated into the proteins, that is PG enzyme. So this normal tomato contains PG gene, then this gene is transcribed into mRNA, then mRNA will be translated into protein enzyme, and that will help in the fruit ripening. So now what is our aim? To increase the shelf life of tomato. So what happens in transgenic tomato? So this PG gene is transcribed into mRNA. We will introduce antisense mRNA, which will complementary to the sense mRNA, right? So that due to the complementarity of antisense RNA, mRNA, that will bind to the mRNA of PG gene, right? And due to formation of double helical, right, that will not get translated into the proteins. In this way there will be no synthesis of polygalactoronase enzyme. So this, if there is no presence of PG enzyme, that cannot degrade the pectin, and the tomato will remain hard. The cell wall of the tomato will remain hard. In this way the tomato ripening is avoided, or you can say the shelf life can be increased. So this is the antisense RNA technology. Once again I will go back to into the genes. Now you know the enzyme polygalactourenates. So how the procedure, what is the procedure? So the gene which encodes for polygalactourenates, that is isolated from the tomato plant, normal tomato plant, then this gene is transferred into the bacteria where the complementary DNA, that is cDNA, is produced, right? Then this cDNA of PG, keep in mind this cDNA that is of PG gene, then this cDNA is introduced into the fresh tomato where that will bind to the mRNA of PG gene, right? And that will there will be formation of double helical, or you can see the sense and antisense mRNA hybrid, and that cannot be translated, keep in mind, and the PG gene or PG enzyme is not produced. So this is all about the antisense RNA approach or FlavrSavr Tomato or antisense RNA technology in the role of this technology in the crop improvement. So this is all about the FlavrSavr Tomato. In next lecture we are going to see how the golden rice is developed. Thank you.